Hello friends, welcome to MedLife. Today we'll be studying about the chapter 2 of pathology, that is cell injury. So first let's talk about pathology. What is pathology? So it is the study of cause and effects of disease or injury, that is changes in cells, tissues and organs that are associated with disease and give rise to the presenting signs and symptoms in patients. Now talking about its etiology, it simply means why disease arises. It refers to the underlying causes and modifying factors that are responsible for the initiation and progression of disease. So what is pathogenesis? It simply means how a disease develops. It refers to the mechanisms of development and progression of disease which account for the cellular and molecular changes that give rise to the specific functional and structural abnormalities that characterize any particular disease. So, etiology means cause and pathogenesis means mechanism of disease. So now let's see about ischemia. So it means lack of blood flow due to thrombosis or atherosclerosis. Now let's come to the cell injury. In the first video of pathology, I have already discussed about all the adaptations, cell injury, reversible as well as irreversible injury. So what is cell injury? If a normal cell experiences any kind of injurious stimulus and the cell is not able to adapt to any kind of adaptations, then the cell will go through cell injury. So now let's see its causes. Number one cause and the most common cause is hypoxia, which means oxygen deficiency, and ischemia. Number 2 is toxins, that is cigarette smoking, environmental pollutants and etc. Number 3 is infectious agents, that is any kind of disease causing pathogens such as virus, bacteria, fungi, parasites and protozoa etc. These can also lead to cell injury. Number 4th cause is immunologic reactions such as autoimmunity and hypersensitivity reaction, in which the cell eats or defects its own cell of a body in autoimmunity disease and hypersensitivity reaction. Number 5th one is genetic abnormalities such as Down syndrome and sickle cell anemia. 6th is nutritional imbalances, in which there is deficiency of nutrients, and excess nutrient can also cause disease, that is cell injury. For example, type 2 diabetes mellitus and atherosclerosis. Number 7 is physical agents such as trauma, temperature, radiation and shock. Number 8 is idiopathic, which means disease arises spontaneously without any known cause. Number 9 is aging. Due to the cellular aging, there is decrease in the normal functioning of a cell, causing cell injury. So now we will study about the morphology of cell injury. So cell injury is of two types, that is reversible cell injury and irreversible cell injury. So if a cell or a cell injury is a mild and transient injury, then the cell will experience or go through reversible injury such as cellular swelling, fatty chains. And if the stimulus is removed, then it can get back to the normal cell. And when the cell injury is of severe and progressive type, then it can lead to irreversible injury, which cannot go back to the normal functioning of the cell. And it is of two types, that is necrosis and apoptosis, which is a type of cell death. So, how to identify whether a cell has undergone reversible or irreversible cell injury? For this, let us see the figure of a normal cell, reversible cell injury and irreversible cell injury respectively, and compare both types of cellular injury with a normal cell. So first, let's see the diagram of reversible cell injury. And find out its characteristic from exterior to interior of the cell. So first, let's see its cell membrane. So here we can see that due to cellular swelling, the cell membrane is intact with the formation of blebs. Blebs is the elevation. Now, second point is endoplasmic reticulum. It has also undergone swelling. Third, ribosome. Here we can see the dispersion of ribosomes has occurred, which means detaching from endoplasmic reticulum. Fourth one is mitochondria. So here we can see that the mitochondria is swelled and there is a small densities in the mitochondria. Fifth point is lysosome. So lysosome is a yellow colored structure which has eaten its own cell organelle, that is mitochondria, and this process of eating its own cell organelle is known as autophagy. And lastly, let us talk about the nucleus. So here we can see in the nucleus clumping has occurred. These are the chromatin, nuclear chromatin. So clumping of nuclear chromatin has occurred in the nucleus. So this was all about reversible injury. Now let's see the figure of irreversible cell injury and compare its characteristics with that of reversible cell injury and a normal cell. So first, here we can see that is there is no swelling, no cellular swelling of the cell. Instead, there is shrinkage of a cell, and this shrinkage is due to the holes or gaps in the cell membrane. So the cell membrane here is not intact. There is presence of holes, that is cell shrinkage has occurred, and because of these holes all the cell nutrients and contents comes out, causing shrinkage of cell. And we can also see that there is no bleb formation as that of reversible cell injury. Second point is endoplasmic reticulum. In the reversible cell injury, we have seen that there was swelling of endoplasmic reticulum, but here in irreversible cell injury, we can see that there is lysis of endoplasmic reticulum with the disruption of ribosomes from the endoplasmic reticulum. Third point was ribosome, which was dispersed. And now let's see the fourth point, that is mitochondria. So here we can see that there is large amorphous densities is present in mitochondria, and swelling has also occurred. Fifth point is lysosome. So here rupture of lysosomes and autolysis has occurred. Now, sixth point is that nucleus. Let us see the nucleus. So here, no clumping of nuclear chromatin, but there are three changes which occurs in the nucleus of irreversible cell injury, and those changes are known as kinosis, karyoresis, and karyolysis. So first let us see what is kinosis. It simply means that is the nucleus becomes more blue and condensed. And karyorexis means fragmentation of nucleus, and karyolysis means fading of basophilia of chromatin. So this was all about irreversible cell injury. Now we will see your table and compare and revise its characteristics again. Now let's see our table of comparison between reversible and irreversible cell injury. So number 1 is cell membrane. In the reversible cell injury, the cell membrane is intact, while in irreversible cell injury, it is not intact due to the presence of holes. And because of the presence of holes, all cell nutrients comes out, causing cell shrinkage. And in the reversible injury, there is the formation of blebs, whereas in irreversible cell injury, there is no such blebs formation. In the reversible cell injury, there is cellular swelling, while in irreversible cell injury, there is no cellular swelling. Instead, there was cellular shrinkage due to the defect in the cell membrane. Now, let's talk about the second point, that is endoplasmic reticulum. In the reversible cell injury, it is swallowed or swelled, whereas in irreversible cell injury, endoplasmic reticulum has undergone lysis, that is bursting of endoplasmic reticulum. Third point is ribosomes. In the reversible cell injury, it is detaching from the endoplasmic reticulum, that is dispersion has occurred, whereas in irreversible cell injury, same dispersion of ribosome has occurred. Fourth point is mitochondria, that is small densities are present in the reversible cell injury in the mitochondria as well as swelling, whereas in irreversible cell injury, large amorphous densities with swelling of mitochondria is present. Fifth point is lysosomes. In the reversible cell injury, there is autophagy process, that is the yellow colored structured lysosomes eat its own cell organelle, that is mitochondria, by the process of autophagy, whereas in irreversible cell injury, the lysosomes are ruptured and there is autolysis. And the sixth point is nucleus. In the reversible cell injury, there is clumping of nuclear chromatin, whereas in irreversible cell injury, there is no such clumping of nuclear chromatin, but there are three major changes which occurs in the irreversible cell injury, and those are kinosis, karyoresis, and karyolysis. So first let's talk about kinosis. What is kinosis? Here the nucleus becomes more blue, that is condensed or compact of nucleus occurs here. In the karyoresis, there is fragmentation of nucleus, that is nucleus breaks into small fragments. And finally, karyolysis is the fading of basophilia of chromatin. So here we finished about the morphology and causes of cell injury. Hope you like it. If you have any question or doubt, please comment down in the comment section, and please like, share and subscribe for more videos of pathology. Thank you.